Hello, my name is Ben Konecny, and my project over the summer at Biochem was to study Protein Kinase A, its two mutant variations, A211D, A211T, and to research how two different mutations in the same residue of PKA result in two distinct diseases with completely different effects on PKA regulation. PKA, or Protein Kinase A, is an enzyme that adds phosphate groups to other proteins, thereby changing the structure and ultimately the function of the phosphorylated protein. PKA is extremely important within our body as it regulates a lot of cellular activity and thus must be kept under control, which is what cyclic AMP does. Cyclic AMP, or cyclic adenosine monophosphate, is a regulator for PKA and directly influences how much present PKA is active or inactive. Issues arise when cyclic AMP either over or underregulates PKA, which could create abnormalities within the body. A few examples of the side effects of over or underregulation of PKA are severe learning disabilities, improper bone growth, and improper muscle growth. These are just a few reasons why PKA is an interesting protein to study. There are many ways to analyze and study a protein structure. I chose to analyze PKA molecular dynamics data using RMSF and Dihedral Angles analysis. RMSF, or Root Mean Square Fluctuation, is the measure of how far each atom moves or fluctuates in respect to the average structure. The backbone of a molecule generally has a fairly low RMSF, while the side chains or outliers tend to have a higher RMSF. The second part of my method was using dihedral angles, which are the angles between two intersecting planes, and are immensely useful when studying a protein's structure and function. Dihedral angles are composed of different types of angles, but the two main ones I focused on were phi and psi torsional angles. With the phi and psi angles, you can graph them with phi on the x and psi on the y-axes. This is called a Ramachandran plot and allows you to easily visualize the changes in dihedral angles over the course of the simulation. These dihedral angle changes can relate to changes in secondary structure of proteins. To perform these methods, I used MD-Trash and PyTrash, which are Python and CPP-Trash packages respectively. Within MD-Trash and PyTrash are the functions that I needed to use, such as RMSF, compute phi, compute psi, and finally plotting, which allowed me to visualize my data effectively. Using these two methods, I compiled several interesting graphs, composed of RMSF, dihedral angles, and Ramachandran plots. This RMSF graph shows the fluctuations of the structure per residue. The residue number is on the x-axis, and the RMSF is on the y-axis, and peaks in two spots, around residue 100, which is tryptophan, and 220, which is glutamic acid. The beginning and end of the RMSF graph can be ignored, as the RMSF will usually always be larger at the termini of proteins. There are regions on this plot where the mutated structures have a higher RMSF than the wild-type structure. Next, my dihedral angles graph shows the fluctuation of the phi and psi angles for each of the PKAs over the course of the trajectory. The wild type fluctuates less than its mutated counterparts. The interesting thing about this graph is that the two mutated residues also behave differently from each other, as you can see here. Furthermore, I plotted various Ramachandran plots, and these showed to me that the wild type had generally less variation in the phi and psi angles, while the two mutants sampled larger conformation areas. My results lead me to conclude that the wild type secondary structure is more stable, while the mutated forms of PKA are more flexible and more likely to fluctuate. Finally, my last piece of data came from visualizing and locating the glycine 80 residue of the A211T mutant structure in PyMol. I found that the mutated threonine 122 was close to the loop that held the glycine 80 and caused the glycine 80 loop to fluctuate and move abnormally, which could be related to the mutation. After performing my dihedral angle and RMSF analysis, I concluded that the A211D mutant PKA is the most flexible, specifically in the regions around residues glutamic acid 100 and tryptophan 220, which is demonstrated through my RMSF and dihedral angle plots. The A211T mutant PKA structure was less stable than the wild type, but more so than the A211D, which was discovered from my RMSF plot. From my Ramachandran plots, I found that the A211T mutant structure varies the most in the region around residue glycine 80, and that the structural region around the mutated residue threonine 122 changes quite less. Upon further investigation on the glycine 80 residue, I found that it lies close to the mutated residue threonine 122 when looking at secondary structure. This influence from the mutated residue threonine 122 could be affecting and changing how the glycine 80 loop moves and behaves. These findings show me that the two mutants behave differently in some aspects even though there was only one residue changed, which relates back to the scientific question that I started with. How do two different mutations in the same residue of PKA result in two distinct diseases with completely different effects on PKA regulation? To conclude, I would like to thank Professor Amaro, the Amaro Lab, BioChemCore mentors, the BioChemCore program, and my peers for making this program an amazing experience and teaching me so much about biochemistry and molecular dynamics, as well as showing me how to do independent research.